Golden retrievers have an uncomfortable relationship with cancer. But when scientists at UC Davis stopped asking why some Goldens die young and instead studied the ones that survived unusually long lives, something unexpected turned up in their DNA. A region involving a gene called ERBB4 separated dogs living roughly 11 and a half years from dogs reaching around 13 and a half, 2 years of life, in a breed where 2 years is everything. And the strange part is that this genetic clue belongs to a family scientists already know very well because of cancer. To understand why that's strange, you have to understand what this breed is up against. In 2018, researchers pulled the records of 652 golden retrievers at a veterinary academic center, dogs whose cause of death had been confirmed by necropsy. 424 of them, 65%, had died of cancer. The most common diagnosis was hemangiosarcoma, an aggressive cancer of the cells lining blood vessels, at nearly 23%. Lymphoid malignancies came second at just over 18%. American Goldens carry an estimated lifetime risk of around 20% for hemangiosarcoma and about 6% for B-cell lymphoma. For a single breed, those numbers are extraordinary. So scientists did the obvious thing. They went looking for the genes responsible. Most genetic studies work that way. You gather the sick, you gather the healthy, and you search for what went wrong. A team at the UC Davis School of Veterinary Medicine ran it in the other direction. Instead of asking which genes make golden retrievers sick, they asked why some golden retrievers were surviving so much longer than others. This is comparative oncology in action, using a species that develops human-like cancers to ask questions we haven't thought to ask in humans yet. Robert Rebin, a veterinary oncologist who co-led the work, framed the reasoning plainly. The assumption is that most golden retrievers carry some genetic predisposition to cancer, but some of them are still alive at 14, 15, 16 years old. If the bad genes really are that widespread, then something in those long-lived dogs might be pushing back. The design followed from the question. Researchers pulled archived blood samples from golden retrievers seen at the university's veterinary hospital, along with samples submitted by owners and primary care veterinarians. They tracked down dates of birth and dates of death, making phone calls and sending emails whenever the records came up short. Then they split the dogs into two groups. On one side, Goldens that reached 14 years or older. On the other, Goldens that died before 12. 304 dogs went into the analysis. Then came the genome scan, comparing those two groups position by position, looking for any spot where the long-lived dogs consistently differed from the short-lived ones. One signal rose above the rest, and it landed on chromosome 37. The gene sitting under it was ERBB4. It goes by another name too, HER4, and that name is the reason this result stopped the researchers in their tracks. ERBB4 belongs to the epidermal growth factor receptor family, a group of receptors that sit on the surface of cells and listen for chemical instructions, then pass the message inward, telling the cell whether to grow, divide, specialize, or hold still. The family has 4 members: EGFR, sometimes called HER1, and then HER2, HER3, and HER4. If you've ever known someone treated for breast cancer, you've almost certainly heard the second name on that list. HER2 is the receptor behind some of the most aggressive breast cancers there are, and entire drug programs exist to shut it down. So a search for longevity in a cancer-prone breed led straight back into cancer biology. And as the study's authors point out, ERBB4 is the only member of that receptor family that can act as either an oncogene or a tumor suppressor. Depending on the setting, it can push cancer forward or hold it back. That ambiguity is part of what made the results so interesting. A gene that can go either way had turned up on the side of the dogs that lived. There was one more detail. The associated variation sat in the 5' untranslated region, a stretch at the front of the genetic message that never becomes part of the protein itself. It's the kind of region capable of influencing how a gene is regulated, part of what researchers studying the field of geroscience, the biology of aging itself, are increasingly focused on. What this particular variation does is still an open question. That detail echoes something researchers had already run into with this breed. Back in 2015, a separate team ran genome-wide association studies on 356 golden retrievers, comparing dogs with hemangiosarcoma and dogs with B-cell lymphoma against healthy controls. They found two shared regions on chromosome 5 that together accounted for roughly 20% of the risk for both cancers, and neither region was explained by a mutation inside a gene. Twice now, in the same breed, on two different chromosomes, 37 and 5, researchers had found important clues outside the protein coding sequence itself, in regions capable of influencing how genes are regulated. One thing worth stating plainly: none of this came out of the Golden Retriever Lifetime Study. That's a separate project run by the Morris Animal Foundation, a long-running effort that enrolled more than 3,000 dogs and has followed them for years, tracking diet, environment, and disease. Different study, different design, different question. It did not find ERBB4. Now the numbers, and these are worth being precise about because they answer a different question than the genome scan asked. The scan compared two extremes: dogs alive at 14 or older against dogs that died before 12. But once ERBB4 surfaced, the researchers went back and sorted the dogs a second way, this time by haplotype, meaning which version of that region each dog had inherited. Golden retrievers carrying two copies of the version labeled haplotype 1 had the longest survival, at 13 and a half years. Those carrying two copies of haplotype 3 had the shortest, at 11 and a half, just under 2 years apart. Across the whole group, the difference was gentler. Simply carrying at least one copy of haplotype 3 was associated with about 11.8 years, against 12.8 in dogs without it. Roughly 1 year, not 2. Danica Banish, the geneticist who co-led the study, put that in perspective. 2 years in a golden retriever is something like a 15 to 20% increase in lifespan. Scaled to a human life, that's 12 to 14 years. Then came the finding that made the researchers lean in. When they separated the dogs by sex, that single copy effect, the shorter survival associated with carrying one copy of haplotype 3, showed up in the females and not in the males. Among dogs carrying two copies, the difference held in both sexes. Nobody has explained why. There are reasons it isn't waved off as noise, because HER4 is known to interact with hormones including estrogen, and it may play a role in how the body handles environmental toxins. But those are places to start looking. They are not an answer. Which brings us to what this study means and what it doesn't. ERBB4 is not the gene that makes golden retrievers live longer. Nobody has shown that this variation causes anything. What the study found is an association in one breed, in one group of a few hundred dogs, at one university. It won't tell you how long your dog will live, it doesn't prevent cancer, and nothing here has been shown to extend a human life. The researchers said as much themselves, calling it one small piece of a much larger puzzle, and their next steps are to repeat the work in a larger population, and to find out whether this variation actually changes how the gene behaves. What makes it worth your attention is the method. Dogs develop cancers that closely resemble ours, and they do it while sharing our homes, our air, and our water, inside a life that runs its whole course in about a decade. That's the premise of comparative oncology, and it's why a golden retriever's genome is more than a curiosity to a cancer researcher. For decades, the search ran in one direction, toward the sick. This time, somebody turned around and looked at the survivors, and the answer led straight back to cancer biology. That's not proof of anything yet. It's something better at this stage. It's a much sharper question.